Very good evening to all of you, and I am really happy to have Dr. Shyam Jee Yadav from Hungary, and he is here for today's session. The session is on antisense oligonucleotides design and its application in gene expression analysis. I hope you all know, most of you have already been attended, already have attended our a few online sessions. And this is one of the programs which we have recently started as during COVID-19 pandemic. Since February, we have been doing free courses, and a lot of them which we have conducted as of now. However, we wanted to do something new, and that is the reason, we are a company which is what I should say, that believe in innovation and creativity. So after 6-7 months of rigorous efforts in providing knowledge and sharing the technology, we thought that let's do something new. And that is the reason why we have launched this program, Envision with BDG. Now, Envision with BDG initiative is a program in which we have asked, or I should say gathered, renowned professionals from all over the world who could share their knowledge and speak, say for 60 to 90 to 120 minutes, say 1 to 2 hours on their topic of expertise. So this is what we have been doing, and today is the fourth session in this row. And I welcome you all the participants for this session, and I hope that the learning will definitely be helpful for your future. So I'll not take much time of yours, and I will just quickly introduce the company BDG Lifesciences to those who are, who have joined for the first time in this session. So BDG Lifesciences is a company which believes in knowledge. And to start off with the slide, we could say that Benjamin Franklin has said that investment in knowledge pays the best interest. And this is the most important point if we understand that why education is so important. So this tells us the story, or I should say the proof, that how much we can reap if we invest in knowledge. So basically, BDG Life Sciences, we are a guide who will help you in solving your academic problems, showing you right path, solving your queries, giving you correct training, paving your path for a successful career, and giving you launchpad to excel in the field of science. BDG Life Sciences was created on the pillars, and one of the major pillars of BDG Life Sciences is that we are a guide, and the motive is to help young students, research scholars who at one point of time, or I should say that at one stage of their life, academic life, I should say, they think that, what I should do next, how I can excel into the field. So the motive of this organization, I hope you know by these 4-5 points, that we are here to show you the right path, solve your queries, and give you correct trainings in the latest technologies, and paving your path for the successful career. And then, as I have already told you, giving you launchpad to excel into the field of science. The next point, if you see, in my slide, if you could see over here, that it's a group. So BDG is what BDG is. Biodiscovery Group. Previously, it was Biodiscovery Group, and now it is BDG Life Sciences OPC Private Limited. Why we have kept group? Now, group is something very important. We all know what is the meaning of group, but then why it is attached with biodiscovery? Now, when we started the company, we started in the form of a group, and the motive was to help people. And that is why when we made this organization as private limited, then we still have not, moved of our thing of helping the people, and that is why it is BDG Life Sciences. So Biodiscovery Group, or I should say BDG Life Sciences, is a company which is being started with the motive to help, and it is for everyone. Until now we have not forgotten our roots, and till now we are helping people in many different ways. So in this slide, you could see that in 2009, we've created a group for students, researchers, scientists, and company professionals who are related to the field of biotechnology, bioinformatics, and pharma. Now, what this group had free consultation, tutorials, books. Back then in 2009, we created a Google group. So at that time, Yahoo groups and Google groups were more popular. Then we had Google Plus, but then now all this thing have been changed, I should say. So we have transformed into different platforms of social media, because now we are on Facebook, Instagram, and LinkedIn. And I would like to show you in our next slides where we are. So this is the motive of starting the organization, and what that motive was: to help. How to help? In the form of free consultation, tutorials, books, project reports, assignments, latest update for trainings, job offers, especially in India as well as abroad, and PhD opportunities or PhD scholarships, and latest science and technology news. So this was the motive of the group. Next, as of now, we have more than 1 million subscribers from all over the world. Now, these subscribers, they are not only students, but they are faculty, researchers, lecturers, scientists, and company professionals. So we started with a small group, but then now we have more than 1 million subscription if we combine all. How to combine all? You could see in this slide that on social media, we have a large number of people who are on Facebook, then LinkedIn, YouTube subscribers, then Twitter followers, and Instagram followers. On Messenger, if you can see, then we have a lot of subscribers on WhatsApp. We have at least 2 platforms of WhatsApp where we are sharing information as well as helping people. In others, you could see we have our own website subscribers where people can go provide their information on our website, and where they can subscribe to our newsletter. And that is how we are, what I should say, that we are helping people. So if you have stuck somewhere, or if you want to know what you can do, then definitely you can visit our website and subscribe to our newsletter, and you can post your queries. So now, exactly, if you want to see that who we are, then BDG Life Sciences, formerly known as Biodiscovery Group, it is a contract research organization that which expertise in bioinformatics, and deals, or I should say provides quality services to the biotechnology, bioinformatics, and pharmaceutical companies and clients. We started, as I've told you, that in 2009 we started in the form of a group to help people, and then in 2010 formally we started the operations, and we started with our research project program, because that is the most important point, that students at bachelor's and master's level, their research profile is not strong. They do not have good publications under their belt, and that is the only reason why why they are not being offered jobs, because our field is research. And when we say research, then it has to be a novel research, it has to be publications, it has to be conferences. So this is the first program which we started, and till now we have completed more than 60 international projects. And people like you, the students, researchers, and company professionals, they have been a part of those research projects. People who wanted to do their major projects or thesis, they have also done these projects and got good marks, as well as international publication, which is an add-on to their curriculum. So our wide array of services include the contract research, workshops, e-workshops, which are online workshops, then research internships, training, webinar, seminar, and I think there was some connection issues. So if you are listening to me, if you can listen to me, then you can just post a message that everything seems fine so that I can proceed. Okay, totally okay. It is fine now. So thank you very much. Let's proceed with the screen sharing and okay. So this is what I was showing you about the program, and that okay, so the research project program, because as we have been doing a lot of research projects, hence this is the bar that is very high. Then we have workshops, on-site and online workshops, where we have been conducting a lot of on-site workshops in India as well as in other countries like Mexico, Iran, Dubai, and Nigeria, Kenya, Sudan, and Malaysia, well as Thailand. So a lot of workshops. Bar is a little up because that has opened the window for. We have been doing online workshops and online training programs since 2012. So we are not new to online mode because people have started online mode since the COVID-19 pandemic. But we have been doing a lot of online workshops and doing it regularly since 2012. Then courses. Now courses, previously, before the COVID-19 pandemic started in March, we used to do at least 2 courses or one free courses every alternate month. But when this COVID-19 pandemic started, in February, from February itself, we started doing free courses, and you will be amazed to know that as of now we have done more than 70 free courses, in which more than 14,000 people from all over the world have participated. So that is something very good for the organization, and it is very good. We've got an overwhelming response. So that is for the courses. And then others, as I have already told you, that we are doing contract research and doing molecular docking services or drug discovery services for our clients. The mission for the organization, or I should say the mission of the organization, is to provide solutions for the future by catalyzing the process of biomedical sciences into therapeutic technologies that benefit human health and society, and eventually fuel economic growth. So this is the motive, or I should say the mission of the organization. These are the services which we are offering And you could see the topics over here: the bioinformatics, basic and advanced, then gene and genome editing using CRISPR-Cas9, Cas12a, Cas13a, and nicases, drug discovery technology, virtual screening, and a lot of programs which we are doing. But I would like to highlight, these are very, these are the old programs, but there are two programs: immune rep set, that is immune repertoire sequencing, and artificial intelligence and machine learning and biology. These are the programs which we started in 2000, in December 2019, and we've got a very huge and very good response. So we've conducted courses and trainings on these two topics, and now we will be launching some research projects on artificial intelligence in drug discovery. Then we have immunoinformatics and vaccine design regularly. Right now also there are two workshops that are currently going on, and one is on bioinformatics, that is for basic, like means people for who do not have any knowledge of bioinformatics, so that is for beginners. And another workshop is right now going on, which is on molecular docking. So molecular docking, we are doing 4 or 5 types of molecular docking, and all the sessions are being conducted on Google Meet, where a live trainer is there to conduct the session. Then last, you could see cancer, HIV, diabetes, and tuberculosis research. So these are the 4 diseases where we, they are saying that I am not audible. So am I not audible, or it is only Ayush Verma? If I am audible, then you can just message that it's fine. Audible, Dr. Sutapa Saha, she's saying that I am audible. So I think if, yes, Preeti Patel is also saying I'm audible, so it's fine. So maybe, you can check your internet connection if I'm not audible. So I was talking about cancer, HIV, diabetes, and tuberculosis research. Now, cancer, HIV, diabetes, and tuberculosis research is something very, we have been doing from a long time. And we have been conducting regular programs targeted on these particular diseases. Like, for example, workshop on cancer molecular targets. Then we have HIV targets. Then we have diabetes and tuberculosis research. So basically, a lot of drug discovery work we have done on cancer, HIV, diabetes, and tuberculosis. So that is the next thing. Now, in this next slide, I would like to point out this last line: innovation, then ideas, then service, and then value. Now, these 4 terms is something very important, or really important to us. Why? The reason being is that we believe in innovation, and that is the only point I would like to give a proof of it late in my later slide, that why we have been kept on, I'm stressing on innovation, because if you will not innovate, then you will not survive into the if you are into the business of technology. Hence we kept on innovating, and then we value, and we kept on taking new ideas, and then creating something new which creates value for you. And that is the reason why I would like to justify that why you should come to BDG Life Sciences for your trainings, for your projects, and for the workshops. Why? Because practical application of technology. We learn by doing the things. When we say that we learn by doing the things, then that is what we give you. That is, we will make you work. We will, in our workshops, in our trainings, or in our research projects, we will make you work, and you will perform the task on your own computer systems. And that is that is how we believe that you will learn in a better way. Then hands-on training sessions, whatever the trainings which we conduct on, either on next generation sequencing, or on what I should say, drug discovery or molecular modeling, they all are hands-on training sessions. Then research projects. Novel research projects means projects on the topics on which nobody else has ever worked before. So that is something very important. And then if the research is not result driven, then definitely there is no use or point of doing the research. Hence, result driven research and everlasting relationship. Everlasting relationship is something very important, and I would like to point out one important point, that people who joined us in 2010 when we started this organization, they are still connected with us. Now, why it would be beneficial for a person who will join now? Because that person who joined with us in 2010 was doing masters. For example, he was a PhD student, but after 10 years that person has become a big scientist, or that person has become a big lecturer or professor. Now that person needs recommendation if he has a job, or if he has some a PhD opportunity. So the person who has joined us now, we recommend that person to him. So it's a circle. Consider yourself, you join now with us, and after 10 years you become a great scientist, and then you ask that, okay, but why you ask? Because you know the level of the quality services which we provide. So you ask us, do you recommend someone? We will say yes, we will recommend. This is the students who have joined us, and you can interview them and you can go ahead. So this is how this entire network works. That is why everlasting relationship is important, and then we will continue to help you until unless you will not stop asking us to help. So that is why this is the slide which I was telling you that I will give you the proof for innovation and creativity, because if we would not have been doing any innovation and we would not have been creative, then this graph would become stagnant. But then until unless this graph is going up of achievements, we are very happy that yes, we are innovating and we are doing something new, and that is why the graph is going up. Just to summarize, we are a 10-year-old company. Trainings and workshops around the world we have done. We have done more than 60 research projects, which included the design, implementation, completion, and international publication. 400 plus workshops around the world, and in India, and 20 plus workshops in other countries. Then we have 50 plus workshops, if you could say, at premier institutes of India like the IIT, the NIT, the VIT, MMC, and JIPMER. Happy customers, you could see the feedback of our people, or I should say participants, who have participated in our training programs and workshops. We have posted video feedbacks, and that is one new thing which we have started 1.5 years back, and you could see the comment of the people on our YouTube channel. So that is all I would like to say. I would like to point out two more important things. We are the first one to introduce online training in drug discovery and designing, and then we are also the first one to conduct the training online and on-site in gene and genome editing using CRISPR-Cas9. So these are some very good achievements of BDG Lifesciences. Free courses, I have already told you that we've conducted more than 70 free courses, and more than 14,000 participants participated from all over the world. So after 6 or 7 months of this hard work, we wanted to give something new, and that is why we started this BDG initiative. Services we are offering: virtual screening, molecular docking, MD simulations and dynamics, predictive toxicology, and NGS data analysis, gene and genome editing. So, and then this is also very important, personalized training. If you have something in mind of which you are not getting the training, then tell us, we will design a program for you and conduct the training. Research project consultancy is especially for the PhD scholars who might stuck at one part or at certain stage of their PhD journey, and they do not do not know where to proceed. So you tell us, and then we will help you in proceeding with your, or I should say, designing the future course of your PhD project. So you can take a screenshot of this particular slide, and we are available. The team is available on Facebook, Instagram, and YouTube, as well as LinkedIn and WhatsApp, where you could ask the questions and they will help you. So I would like to first of all thank you for listening to me, and then now I would like to introduce Dr. Shyam Jee. He is from right now he is in Hungary, but He is a postdoctoral researcher at the Laboratory of Cellular Imaging and is working as a research associate at Institute of Biophysics, Biological Research Center, Szeged in Hungary. His work is on drone-based biosensor development project. So he has joined as a researcher in 2015 at the Institute of Plant Biology, Biological Research in Hungary, and proving his academic excellence, he has won the EMBO EMBL Fellowship Award at the EMBO EMBL Symposium in Germany. So we have a very renowned speaker with us, and now I would like to what I should say that I would like to hand over to Dr. Shyam Jee, and so that he can continue with the session, and I hope that it would be fruitful for you guys. And Dr. Shyam, now it is up to you. You can unmute yourself and please go ahead.
Okay, could we share the screen?
Yes, hello? Please go ahead. Yes, Dr. Shyam, you can please go ahead.
Is it visible for everyone?
Yes, it's perfect.
Okay. Sorry. Okay. Thank you very much for your kind introduction, and it was really nice to hear about whatever the services that you are providing, especially the young students or who is looking for the who is looking their career in the biological research. So it is really nice. Even I was also there, so I know what is the value of these things, that if you are doing graduation or something like basic level, so these kind of the training are really helpful. And if you want to think about like to do something like advanced research, so everyone is looking that, do you have any training? Do you have got any experience like handling pipette or doing something? So most of the persons are researchers who is well settled. They are looking those kind of students who can handle these kind of things. And whatever the services that you are providing, that is really nice. I appreciate it. And thank you very much for inviting me for this presentation. I hope this presentation will be helpful to all of you. And if there will be any questions, so I'm available here. So there is my email ID, that is sam.jee@brc.hu, and it will be also at the last this email ID. If there will be any query regarding to my works or for any suggestion, if you want to contact me, so it is okay for me. So mostly if I will be free, then I can reply on that time. Otherwise later I can reply to your questions. Okay, so let's coming on my presentation. So today's I'm going to present like antisense oligo designing and its application in gene expression analysis. So here we can see there is two part of my presentation. One is antisense oligo designing and other is its application in gene expression analysis. So actually this work performed by me during my PhD study. So that's why I'm going to talk about it, and I am hopeful that it will help you, or it will give some idea that how we are doing silencing by using this antisense oligo. So just basic, what is the antisense oligo? Actually, antisense oligo, it is the single and short standard synthetic DNA molecules, and that is typically 15 to 25 nucleotide long, and it can bind to mRNA, target mRNA. So that is designed for that gene, and it can lead the gene silencing by different ways, such as by forming here such as by forming the heteroduplex like antisense oligo with mRNA, and it will inhibit the translation, and in another way like by the activation of RNase H enzyme, so there will be cleavage of mRNA. And here it is important that this antisense oligo will lead the cleavage of the mRNA, but this will be remain intact. So this antisense oligo will be free to interact with other mRNA molecules. So in this way, one antisense oligo can interact with several mRNA molecules. And it can also, it is also able to inhibit the ribosomal assembly, and these are the activity that will be occurring in the cytoplasm. But this antisense oligo, it can also enter into the nucleus, and where it will regulate the gene expression by inhibiting the cap formation at the 5' end in the mRNA, and also by inhibiting the RNA splicing, like here, if that antisense oligonucleotide binds to the exon and intron region, so it will inhibit the splicing, and it can also activate the RNase H enzyme for the fragmentation of this target mRNA. So in this way, this antisense oligo is important for the gene silencing. We can choose this antisense oligo to silence any of the genes and to see the effect. So this is usually used for the loss of function analysis. So we are inhibiting any particular genes, and we are going to see what is the output of the inhibition or silencing of that gene. And the antisense effect was first time, the effect of antisense oligonucleotide was identified by Zemnick in 1978. So here, this is the reason that you can see in the nucleus how these are the reasons that can be inhibited by the antisense oligo. So now we know what are the importance of antisense oligo, but how to design antisense oligo to increase the hit rate or to increase the silencing effect. So there are some important parameters. So that is really important to design the effective antisense oligo. So like here, prediction of the secondary structure of the RNA, motif search, identification of preferable RNA secondary locally structure, GC content calculation, and binding energy prediction. So these parameters are important to design the effective antisense oligo. So I will be talking one by one and how we can increase the silencing effect. Sorry, this screen isn't moving. Okay. For the production of the secondary structure, so there are two programs that is online available. Anyone can use these two programs. So one is S-fold program, other is M-fold program. This S-fold program used for the production of the secondary structure. And whereas this M-fold program, this is also used for the production of the secondary structure, but it also calculates the overall free energy for the binding capacity. So we can know what are the energy that is required for the binding. So this sequence here, we can see this is the secondary structure of a particular RNA. And it was designed by using the M-fold program. And this arrow indicating that these are the regions that can be used for the antisense oligo designing. But there are several factors, several other parameters that will decide which one is the best one. So we will see what are the other parameters. So here, this is the suggestion by the Matvira et al. So based on their research, so they suggested there are some motif that is showing positive effect on the antisense oligo. So that is here in the green color. And there are some other motifs that have negative effect on the antisense oligo. Suppose you decided some reason for the designing of antisense oligo that is also following the secondary structure criteria, but there is like a more number of this red color motif. So we will not choose that region for the designing of antisense oligo because they have weak effect. So we will only consider the region that have the green letters, that is green letters, that is showing the strong effect. So here there is one genes. Here we can see the motif search was performed. This red color, that is weak effect, and this green color, that is indicating the strong effect of the antisense oligo. This yellow color backgrounds, this is indicating the reason that can be used for the designing of antisense oligo. So they are the different reason that can be used for the designing of antisense oligo. So after motif searching, so now we will be looking for the identification of preferable RNA secondary local structure. So suppose we saw previously that there are some regions that can be used for the designing of antisense oligo, but if we are looking for the local secondary structure, so we will search, is there hairpin formation at least 3 base pair? If there is 3 base pair or more than 3 base pair, there will be hairpin formation. So like here in down, we can see there are the 4 nucleotide that is showing the base pairing. So this reason cannot be used for the designing of antisense oligo. Only 2, like 3 or less than 3 base pairing is allowed for the designing of antisense oligo. But if there is more than 3, so that will be discarded. That region will be discarded, and that will not consider for the designing of antisense oligo. And after that, there is other parameters like GC content calculation. So suppose we selected one region that was following the criteria of secondary structure and motif search, and after that we will determine whether this selected region have what is the GC content. The GC content, G and C both, if that is more than 55%, so that will be good for the designing of the antisense oligo, that will show the stronger effect. But if there is the GC content is like less than 55%, here let's talk like here is the 11 GC content and there is just 980. So this is the more than this is 55%. Okay, so this reason can be considered for the designing of antisense. And if the GC content more than 55%, that is really good for the designing of the antisense oligo. But minimum there should be at least 55% GC content. And here is the, so here is the like total energy that is for required for the binding, and this antisense mRNA binding, that is the required energy. And here we can see the reason that was following that criteria. Like this is the reason that like GC80, and here upper region that is like indicating the number. So this is our target gene, and here the designed antisense oligo that is like showing from 2. Here also there is a numbering here, if you can see, like 22, okay, 2210. So this is the reason. From that reason, antisense oligo designing is start, and it will go up to like in this case it was 20. So from 2210 to 20, like it will go up to 30. So it will be that will be the reason that will be designed. So in case the software here indicating that this is the designed antisense oligonucleotide. So this is the final step. If you process all the criteria like secondary structure search, motif search, GC calculation, and after that, so this is the designed antisense oligo. But antisense oligo can be degraded by the nucleases. So to provide the stability into the antisense oligo, so usually there is a different kind of modification that researchers are following. So there are the three regions that can be modified to provide the stability of designed antisense oligo. Like here, we can modify the base of nucleotide, or we can modify it on the ribose, and or we can modify the phosphate backbone. So one of them can be modified to provide the stability. And someone can think that why not we can provide twice modification like in two regions. That is not necessary. Just provide the stability. Just one modification will be uh, sufficient. And usually researchers are providing the phosphate backbone, and based on this modification like base, ribose, and phosphate, there is also like generation, like kind of generation, like this is the first generation chemical modification that considered as a phosphate backbone. If someone is doing here, that will consider as a first generation. If with ribose, that will be coming in the second generation, and in base, so that is coming in the third generation. So based on the stability and advantage, so researchers are like categorized this modification into three generation: first generation, second generation, and third generation chemical modification. So now third generation, like in base. Usually this antisense oligo is taken by the cell through the endocytosis process, but there is also some methods like that can increase the uptake of antisense oligo. So like mechanical techniques like electroporation, microinjection, and particle bombardment. Especially in the medical field, this microinjection is most commonly used method, and this cationic lipid carriers. So there is also like cationic lipid that can be used to increase the uptake rate. So this cytofactin, mostly the plant biologists are using. Even I was also using this cytofactin, and I was also using this particle bombardment like gene gun method. So these are the methods that can help you to increase the antisense oligo uptake into the cell. And there are many medicines available by using the antisense oligo. There are some examples like fermivircin, so that is used against the peripheral cytomegalovirus retinitis AIDS patient. And there is also oblimercin that is used for the various type of the cancer, and ixodynasin 51, and that is market name is like iteplersen. So that is used against the like Duchenne muscular dystrophy, that is especially caused by the specific mutation in the gene, and nusinersen, and this is used against the spinal muscular atrophy, that is also a rare neuromuscular disorder. So, and there are also some antisense oligo that is used in the clinical field. So this is the use of antisense oligo in the medical field. But how we can use in the plant? Actually, there are not much research happen in the case of the plant, but there are some articles that is talking about the application of antisense oligo, especially silence of some specific genes. So we decided to use this antisense oligo for the silencing of our interested gene. So we used this antisense oligo for our project. So now I will describe that how we decided to use, and what was the process that we used during the silencing, and what was the output of that project that we were involved by using this antisense oligo. Okay, so here in the normal position, normally if there is pollen coming on the stigma of the stigma, so it start to germinate the pollen tube, And the pollen tube of the pollen is going in a long way, and it is going to the embryo sac through the micropyle, and where one sperm cell fertilized with the get fused with the egg cells, that is making the zygote, and other sperm cells fused with the polar nuclei and forming the synergetic cell, and this synergetic cell providing the nutrient for the developing embryo. So this is the normal situation, or normally it is occurring in the nature. But there are some factors that can influence the function of the, that can influence the function or the growth of these pollen tubes. Like there are several factors, maybe there are physical factors and genetic factors. In physical factors like salt stress, salinity, heat stress. So there are different kind of the physical factors that can affect. But there is also some molecular factors that can influence the pollen tube growth. If this pollen tube growth will be inhibited due to the any reasons, so there will be not any fertilization. If there isn't any fertilization, so the formation of seed or fruit will be not occurring. So in this case, we used our genetic factors. How this genetic factors, so we know there are some genes, they involved in the pollen tube growth, and especially the polarity formation and tube elongation. So there are some small GTPase proteins, and the RAS also belong to the small GTPase proteins. So in the case of plant, here we can see there is the ROP, and in the case of animal, there is some other small proteins, small GTPase like proteins. So our focus was to focus on the ROP because we were working with the plant system, and ROP have different regulators and effector molecules that can interact with the ROP and that can regulate different kind of the physiological function. So here we can see ROP GTPase are inactive state in the pollen, but once pollen is start to germinate, so ROP are starting to accumulate at the growing region, and here we can see the more accumulation of the ROP. ROP have several effectors also. ROP itself interacting with several genes, like one of them is receptor-like kinases. So receptor-like kinases, they interact with the ROP GTPase and they regulate some physiological function during the pollen tube growth. And they involve in polarity and pollen tube growth. But same like receptor-like kinases, there is also some other kinases. They don't have like receptor on the cell surface, but they also interact with the ROP. So here we can see RLCK, that is receptor-like cytoplasmic kinases. They are cytoplasmic kinase, but they are not a receptor. They don't have the receptor. So these kinases are also interacting, but the function of this receptor-like kinases were not known. So that's why we decided already in our group, they have discovered that how this RLK receptor-like kinase interacting with ROP GTPase and how it is regulating the several function during the pollen tube growth. But this receptor-like kinases, in this regard, there wasn't any work. So that's why they assigned this project to me to identify the function of receptor-like kinases. So here we can see this ROP GTPase. If it is interacting with GTPase, so it is active state. If it is interacting with GDP, so it is inactive state. And in active state, in the active state, ROP GTPase regulate different function. You can see actin dynamics, exocytosis, calcium ion channels, and also NOx. And NOx is regulating the ROS. And ROS is really important for the rapid growth of pollen tube. So our aim was to identify those RLCK6 kinases of Nicotiana tabacum which are expressing in the pollen tube, and designing a synthesis of a specific antisense oligo against the each pollen-expressed tobacco RLCK6 kinase, and investigating the pollen tube growth and morphology in relation to the application of synthesized antisense oligo. So Arabidopsis thaliana gene already sequenced. So in database, we can find the Arabidopsis thaliana genome. But in the case of Nicotiana tobacco, in that time, in 15, so there wasn't like, that sequence was not whole sequenced. So there was some predicted sequences by using like in silico method. So we used that predicted sequences of RLCK kinase and this Arabidopsis thaliana, that sequence data. And here odd number are indicating that like Arabidopsis nucleotide, and even number is indicating like Nicotiana tobacco. So we did homology search by using cluster omega tool. So here we can see the homology between these two nucleotides, these two different species nucleotide, and this yellow background indicating that conserved region between these two species. And based on this homology search, we prepared the phylogenetic tree, because already we know the sequence of Arabidopsis thaliana, but we are not knowing the sequence of Nicotiana tobacco. So based on this homology search, we prepared the phylogenetic tree. Here we can see the results. The name of Nicotiana tobacco was given like the sequence that is showing the its closest homology with the Arabidopsis thaliana. Here we can see this is the Arabidopsis thaliana kinase 1, and this is other reason that is showing the like closest relationship with this. So it was named like 1. So this is Arabidopsis thaliana kinase 6A1, and this is also 6A1, but this is the Nicotiana tabacum gene. Okay, so like this, based on this closest homologue, so the sequence of Nicotiana tabacum were named. So here we can see like Nicotiana tobacco A2 and Arabidopsis thaliana A2. So like that way, so we named the sequence of Nicotiana tobacco based on its closest homologue. So based on that conserved region that we found after this homology search, we designed the primer, and after that we checked the expression pattern. We know the expression pattern of Arabidopsis thaliana, but we were not knowing the sequence of Nicotiana tobacco. So that's why we used that conserved region to design the primer, and we did the PCR analysis. So here we can see the expression pattern of RLCK6 in the Nicotiana tabacum. So here we can see we used like pollen, germinated pollen, and leaf and H2O as a control. So in the case of A3, A4, and A6, here we can see in the pollen. So there is strong expression of A3 kinase in the pollen, and A4, that is also a strong expression in the pollen, and A6 also. And here A7 is also showing some weak expression, but there is expression. And other kinases like A1, there isn't any expression in the pollen. A2, there is very weak expression, and in the case of A5, there isn't any expression. So we found that A3, A4, and A6 were strongly expressing in the pollen, whereas A1, A2, and A5 were expressing in the leaf. But we selected only those regions that were expressing strongly in the pollen. But in the case of this Arabidopsis thaliana, there was prediction, there was also like expression pattern analysis. So that result was showing that A7 kinase was expressing strongly in the pollen. So that's why we decided to also consider this A7 because there is also some expression in this case. So we decided to use A3, A4, A6 and A7 for the designing of antisense nucleotide. But as I mentioned, there wasn't the known genome sequences. So we just only did homology search and based on conserved region. So we did PCR analysis. After that, we isolated the mRNA and we sent it for the sequencing. So after getting the sequencing results, then we design the primer. So for the designing primer, first step we followed, like we did the secondary structure analysis. So here we can see the secondary structure of one of our kinase like A3 mRNA. So in this arrow is indicating that the region that can be used for the designing of antisense oligo. And in the same way, the other kinase like A4, A6 and A7 secondary structure was also performed. And this was performed by using the M-fold program. And after secondary structure, we perform motif search of A3 kinase. So in all the cases, to represent the picture, so I'm all the time considering just only A3 gene. So in a same way, other gene was also performed. So like here we can see the motif search, like the red color letter that is indicating weaker effect for the antisense designing, green color that is showing stronger effect, and this yellow color background indicating that the reason that can be used for the designing of antisense oligo, and this green color background indicating, so this was the reason that we used, especially for the A3 kinase for the silencing purpose. So in a similar way, other was also performed. So to see the silencing effect, so we were growing our pollen into the liquid media and we applied into the liquid media, and this pollen while growing, they can take the antisense nucleotide from the media through the endocytosis process, and to increase the uptake we can also use like cytofactin. So in our case, we use the cytofactin to increase the uptake of antisense oligonucleotide. So once we observed our results in the microscopy, so here you can see normally the pollen is growing like in a straight way. But once we applied the antisense oligo that was silencing for the kinase, so the silencing effect of kinase are causing branch formation. So this is unusual case in the case of normal pollen growth. So once we calculated this branch formation, so we found that okay, there was also sense control. So like sense stand we also designed to see whether this sense also can cause any harmful effect or they are also just like the control. So there isn't any effect. And in the case of this treatment, here we can see the branch formation was like going around between 16 to 18%. So it is really high number if you are considering the, if you are comparing with the normal, because in the case of normal there isn't branch formation, but in the case of treatment there is branch formation. So this is indicating yes, this kinases have some effect on the branching. And there was also some different kind of abnormality, like here you can see the knot formation. This is like bubble in the middle of the pollen tube. So normally pollen tube is just like cylindrical shape, but there isn't like bubble or branch formation. Once we calculated the ratio of this knot formation, so it was again going around 10%. Okay, so these are the different genes that was silenced separately. So they have different effect, like some genes showing a stronger effect and some gene showing the weaker effect. Here, if we are remembering that the expression pattern analysis, we are able to see less expression in the pollen, but in a certain phase of pollen tube growth, this A7 is playing important roles. So there is some more knot formation if you are comparing with other gene like A3, A4, A6. And the third kind of the abnormality that was observed, like here, like widening up the tip. So this was the less, not like branch formation, but there was also this kind of like widening of tip. So we calculated the ratio. So it is going around 6% and control is less than, okay, it's around 1%, but in the case of treatment, it's going around 6%. But there are some stronger effect in the case of A3 kinase, but weaker effect like in the case of A6 and A7. So in conclusion, we found that the 4 members of RLCK 6A family were strongly expressing in the Nicotiana tobacco pollen, and these kinases were selected for the antisense oligo designing. The partial silencing of RLCK 6A kinase member was attempted using designed antisense oligo taken up into growing pollen tubes. And the observed phenotype treated pollen tubes supported the view that these kinases are indeed involved in rope GTPase mediated polarity establishment and tip growth process. So in conclusion, in overall, if I can summarize this presentation, especially the use of antisense oligo in the plant, if there is a rope GTPase, like if there isn't any mutation or isn't any silencing in the case of rope GTPase, or their interacting partners like receptor-like kinase or receptor-like cytoplasmic kinase, if all the genes they are working normally, so there will be normal pollen tube growth. Here you can see there isn't any like widening up the pollen tubes or branching up the pollen tubes. So there isn't that kind of abnormality. That is just normal pollen tube. So if there will be normal pollen tube, so that will lead the fertilization and that will lead further the fruit and seed development. But if there is any problem with like rope GTPase or in RLK, like receptor-like kinases or receptor-like cytoplasmic kinases, so that will lead abnormal pollen tube growth. And this abnormal pollen tube growth will be not able to fertilize the embryo. And if there will be no any fertilization, so the chance of fruit or seed development will be there. There shouldn't be any chance to get the fruit or seed from that event. So in this way, I conclude my the presentation, and at last I would like to thanks my supervisor Professor Attila Fair and co-supervisor Ildiko Walkei and Dr. Farhan Ayadin and Ildiko Walkei, and they were helping me during the microscopic works, and they taught me how to use this fluorescence and confocal laser microscopy. Especially there were different kind of microscopy. And Dr. Georgie Ferenc, she taught me how to design antisense oligo, and she also gave me the software. So during my PhD, I was doing my own works. Just only synthesis part was performed by the Georgie, and the remaining part I was doing for myself. And currently I am working in this group with Dr. Farhan Ayadin in his group for the biosensor development. And these are the funding agency like National Research Development and Innovation Office. And I was the scholarship holder, like Stipendium Hungaricum scholarship. So my PhD work was especially financially supported by this Stipendium Hungaricum scholarship. And I would like to thank you to everyone for listening this presentation. And if you have any question or any doubt, so you can ask me in the chat or anywhere, I don't know, maybe live also. Thank you. Thank you for your attention.
Thank you, Dr. Shyam, for your enlightening session, and I'm really thankful to you that you have taken out your, this time from your valuable schedule. And I am definitely, I'm sure that this will definitely help the participants in gaining knowledge. Now we have got 2, 3 questions. So, Dr. Shyam, I would like to just address these questions.
Okay.
Uh, First is from Dr. Sutapa Saha, and she said that is Edit Lower Primer the software platform to design antisense oligo?
Okay, may I go there?
Yeah, it's in the chat if you can just see.
Okay. Chat. I couldn't see where is my chat.
No, you just have to open Zoom and then click on chat.
Yes, actually, I'm looking for that. Usually, okay, maybe here hidden.
Yes, we have seen, and the feedback link is already being posted into the chat box. So you need to just click on it, and obviously someone is saying the password. The password would be your Gmail because you have to sign in to give the feedback. So feedback link, it is not working. No, it is working. The feedback link is working. I have just clicked on it, and it has
Okay.
directed me the
Actually,
feedback.
I couldn't find that chat box. Unfortunately, it was here, but I don't know where it is.
Okay, so no issues. We can email. I will tell
Okay,
you the.
just
Uh,
could you repeat the question?
the question is, even I could not understand, is Edit Lower Primer the software platform to design antisense oligo? So I think she is asking, Dr. Sotapa is asking about the software platform to design the antisense oligos.
Actually, in the presentation, you can see there is like two, like upper and a lower region. Okay,
Mhm.
but once we are putting our like sequence, so the software automatic that is giving. So
Yeah, which software I think she is asking.
yeah, so that software automatic, we are not doing any things like manually, just that is coming into the software.
Okay. So no. I'm just her question is that which software platform, like whether you are using Galaxy
Oh,
or
sorry. Yeah, actually, yes. I used Oligo 5 program. Okay.
Okay. Dr. Sutapa, I think now you can see he has answered. Oligo,
Oligo 5 program. Actually, that is
oligo.
not freely available. So, especially in our institute, there is a one group. They have like all the software, whatever the required, and that is the paid version. But she gave me, because I am also the part of that institute, so she gave me. So that's why I was able to use that software.
Oligo 5 program. Now we
Yes.
have one more question that they, from Nancy Jaiswal. So she asked that she wants to know the tools for calculation of GC content and binding energy. Now, however, I would like to interrupt you, Dr. Sham, over here because
Uh,
GC content, if we need to find, then we can easily find it from the fast queue, right? Or multi QC, or they both are clubbed in Galaxy,
yes.
right? Am I right?
Yes.
Yes, and then for binding energies, I think she is referring to the binding energies of which we get after molecular docking. So there are a number of softwares for it, like paid softwares are there from BioVia, Discovery Studio,
Actually,
then
yes. Yeah, there is also RNA 3.5. That is also a software that can be used for the like to know the exact binding energy. Okay, there is separate software. Yes, I know.
definitely.
Yeah.
And then someone asked, what is ROP?
Oh, that is the row of plant. Like I saw in the beginning, there is like some small GTPase like proteins. So in that there is like plant family, and in the yeast and in animal. So that is also just like similar the like row in the animal, but in the row in plant that is similar, but in plant we don't call them row, we call them row of plant. That is
Okay.
the row, but row of plant. So
Okay.
row of plant, so that is the row.
Okay. So,
Hmm.
and then one the question is from Ademi Kayode. And I think the question is, what is the best method of delivering ASO into cells?
Actually, we cannot say which one is the best, but in the medical practice, usually doctors are prescribing or using the micro injection. Okay.
Okay, okay, okay,
But
okay,
in the case of plant research, okay, I saw there are some articles, but all the time they were using just cytofactin.
okay.
But
And
but
one
they
more, I think I
yeah.
will take, this is the last question.
Hmm.
They are saying that you said that antisense oligonucleotide will not allow splicing of the mRNA 5' capping. So
Yes.
how designed primer which will cause silencing more specific to the silenced work of 5' capping and 5' capping and silencing?
Sorry, could you repeat once more?
Yeah, it's a very long question. I think.
Just only, I just maybe just only just once you repeat it, I will catch it.
Yes, sure, sure, sure. How the design primer, which will cause silencing, more specific to silence the work of 5' capping and silencing.
Okay.
If I could not understand the question, but
Actually, suppose, okay, so there is a one gene. In that gene, there is some reason like there will be five terminal or okay, five five prime or three prime terminal region. So that gene will contain some sequence. If we are focusing while designing, if we are focusing at the five prime region, okay, if five if that antisense is going to bind at the 5' region, especially on that region, because this is nothing, just complementary sequence. And this can bind only those regions that is the complementary to this. If we are focusing while designing on the 5' region, like especially our gene, we are designing. But if we are designing at the like 5' like beginning regions, so that will definitely that will focus only on that region. So in this way, while designing antisense oligo, we are all the time considering which region we want to like silence, middle region, terminal region, or we can decide which region we want to silence. And that antisense oligo will bind only that region because this is the specific sequence.
okay,
and uh,
okay,
and one more things, after designing we are performing all the time, we are performing like blast search whether this sequence is going to bind other genes or not. If this is going to bind other gene, so in that case we need to discard that region, we will not consider that region. If there will be like very less homology with other gene, and that is like considerable, like less than 25%, then we can go with that primer or with that reason that can be used for the antisense. If that is showing more homology with other genes, suppose we are silencing kinase A3, but it is going to silence some other gene, so that will be not useful for us. So in that way, after designing, we need to go for the blast search, and there we will decide which region or which one we are going to use for our work.
okay, Dr. Sham. Thank you very much for your time and thank you for your explanations. And I hope that people you would have enjoyed it. The feedback link is already been posted into the chat, and it is working. However, you need to, because that's the Google and we have limited to one response, so you need to sign in and then only you can go ahead and provide the feedback. Now the timing or the deadline to provide the feedback is tomorrow India time 1 p.m. So by that time the feedback link will be accepting responses, and after that the link would be disabled. So you have time till tomorrow 1 p.m. to provide the feedback. Tomorrow we have another session, and the session would be very exciting because it is on developmental biology by Dr. Naga Girish Boshnala, and he is the dean from Manipal, Bangalore, India. So Manipal University. So if you want, you can go through the website and you can register for it. That is also a free session. And in the chat box, we have already posted the our web, the link of the website for upcoming events and hands-on training sessions, which are 5 days or 7 days and 6 days. So you can go and you can register for it according to the the technical skills which you want to learn. And thank you very much for all, from all the participants. And Dr. Shyam, thank you very much for your time and for giving us a very enlightenment presentation. Thank you very much.
Okay. I also thanks to everyone for the, I thank to you that you have given me opportunity to share my research work, and all the participants that they were listening my presentation. So I am thankful to all of you.
Leave the meeting all, and we will be ending the meeting.
Okay.
Thank you very much.
Bye.